Hi, I'm Jake Byrnes. I'm a co-founder and chief scientific officer with Barnwell Bio. This is a re-recording of a talk I recently gave at the 2026 Poultry Science Association meeting. I was part of a symposium that was talking about litter management. Title of my talk today is Comprehensive Proactive Biosurveillance, Tracking DNA and RNA Pathogens Using Shotgun Metagenomic Sequencing of Environmental Samples. Very quickly, here's a talk outline. What motivates my talk today is really talking about the challenges posed to our poultry operations in the US by infectious disease. I'll talk a little bit about current best practices for monitoring, and then I'll get into an approach for using proactive biosurveillance. I'll talk a little bit about environmental sampling and some of the target agnostic tools that we can use, including multiplex PCR and metagenomic sequencing. And then finally, ways to think about integrating a proactive biosurveillance approach with existing practices. Last but not least, I'll talk a little bit about a case study where we can show how we've applied our platform and about the future of some of the stuff that Barnwell is doing with developing viral RNA metagenomics. I don't think I have to tell any poultry-based audience this, but obviously infectious disease is a huge problem for the industry. It can take days, weeks, and sometimes even months to diagnose an outbreak. And as a result, globally, there's estimates that up to $300 billion are lost in production each year due to infectious disease. The most obvious or salient example of this is this current avian influenza outbreak that's been ravaging through the US since 2022. Latest estimates say that we've lost up to 206 million birds that have been culled due to this avian influenza outbreak. And there are estimates that about $1.4 billion have been paid out from the federal government for outbreak response and indemnity payments. And just, some quick quotes from folks that we work with. Some have called it Operation Doomsday in dealing with these outbreaks. And in general, it's just so frustrating when disease is affecting your livelihood. So infectious disease is a very big problem. I don't think I have to say more on that. Um, when we look at kind of standard monitoring today, it's comprehensive, but importantly, it relies a lot on the observation of clinical signs. And so, sort of typical health monitoring or management practices today, managing and collecting signalment, so kind of standard basic barn stats that get recorded every day, including vitals and also sort of production metrics. Um, Also, there's common flock examination. In this particular report that I'm referencing from, they talk about visual inspection, nose to toes, audible smell, touch, all the kind of observational cues you can pick up in a barn. And often these can be sort of your first indication that there's something wrong. And of course, we also post birds and perform necropsies to identify those kind of hidden symptoms in sick birds. But I would argue that waiting for symptoms to appear misses this kind of earliest opportunity to identify a disease because often things are in the house before we even know it. So this is just a cartoon for how this works today. Kind of the current state of health monitoring is really reactive. So in the middle of this graph, hopefully you can see my mouse. I'm showing kind of a standard progression into an outbreak. You have sort of a healthy barn. You have those sort of earliest pre-symptomatic cases of an infectious disease. Eventually, this spreads more and you have more and more infected birds, and you start to see symptomatic cases and eventual outbreak. Traditional practices really don't pick up on anything until we get into this last part of this workflow where you start to see symptomatic cases. Then there's an opportunity to apply diagnostic testing, identify what's going on, and then intervene. And what we propose is if you have a proactive continuous surveillance system in place, there might be an opportunity to catch things a little bit earlier. So here what we're showing is imagine if you're testing, every few days or every week or every couple weeks in your house, and this kind of lower graph is sort of showing, each one of these green spots are kind of healthy results from that proactive biosurveillance. And then hopefully there's an opportunity that we start to see our early kind of suspect results coming during that pre-symptomatic period of the infectious disease spread. If we get these suspect results early, we can do confirmatory testing earlier, and then hopefully get to identifying and intervening to stop spread sooner. So, not surprisingly, taking a proactive approach to biosurveillance is not all roses. It creates new challenges. Most importantly, in the absence of those clinical signs, there are kind of two major challenges that arise. The first one is which animals should you be sampling for your testing? Traditionally, animal sampling for necropsy or diagnostic testing is driven by those symptomatic animals. When you're thinking about who to test, you look for the animals that look sickest. Without those clinical signs, there's nothing really guiding your animal selection, and it more or less becomes a numbers game, right? The more animals you test on, the higher your odds for detecting something in the house. But as we know, a lot of individual diagnostic testing and sampling is very labor intensive. In addition, the other kind of major challenge is similar. It's, which infectious agents should we look for? Typically, we use those clinical signs to help us kind of narrow down and identify, one or a handful of suspected pathogens that might be causing what we're seeing. But again, in the absence of clinical signs, anything could be circulating in our barn and we don't know. And so, to make proactive biosurveillance effective, we really need an approach that's holistic where we can test for all pathogens simultaneously. Um, First of all, trying to attack that first problem, trying to solve which animals to test. This is an opportunity to actually start to use environmental sampling. And the general premise here is that, the entire flock is continuously and passively contaminating the entire barn environment. And, taking environmental samples allows us to get kind of a barn-wide sample. And, that barn-wide sample hopefully is picking up signal from the entire flock. Here, I'm just showing a couple examples. There are many other approaches to environmental sampling, but a few key ones. You can take litter grabs, and litter primarily contains feces, feathers, skin, maybe some respiratory stuff as well. They're easy to collect, but they have limited spatial representation. It depends on how many places you sample. Um, Boot sock swabs are a really good source and one that's been used quite a bit by the industry. Again, typically you're walking those swabs through litter, so it's sort of picking up the same matrices. The advantages here is it aggregates signal across a large floor area. Depending on how much ground you cover, you can actually pick up signal across the whole barn. Um, it doesn't get into things below the litter surface, which may be important if there's things kind of growing underneath. But generally covers good ground, and there's been a lot of studies to sort of show that it's generally more sensitive than litter grab samples. Another option here is sampling water sources, right? You can sample drinkers, or you can actually sample egg wash water in the case of breeders or layers. And so, again, this is kind of an interesting source because it might cover different matrices. You might actually have a better chance of picking up some oral or respiratory pathogens if you're doing drinkers, or possibly, get closer to cloacal infections if you're looking at wash water. Um, Let me continue. Talking about kind of that other major challenge around, how do we know what targets to look for? There are a number of technologies that are target agnostic, partially or sometimes, they cover the kind of whole comprehensive ground. And the nice thing about these is we're getting to a space where it sort of removes the need to know ahead of time which pathogens you're looking for. Again, there are more out there, but here I'm kind of highlighting a couple key tools that are in the toolkit. One is multiplex PCR, and the other is using shotgun metagenomics. Multiplex PCR, generally you can cover, 10 plus targets. So it's got decent target breadth. Some of the advantages, it's very sensitive. It's generally quick and relatively cheap. and can be fairly high throughput. Some of the disadvantages or challenges, it requires specialty lab equipment and trained personnel. You have to do individual probe design for each target, so there's some linear spin up time as you add more targets. Shotgun metagenomics, which Barnwell relies on, is a really interesting approach. It's a more recent development in the field. One of the key powers of it is its breadth. You can simultaneously detect all DNA and RNA-based organisms in the barn. So you can really cover all the ground you want to: viruses, bacteria, fungi, parasites. It has the ability, if you sequence deeply enough, to get down to strain level resolution, so even below species level, and really kind of figure out what pathogens you're dealing with. You can also do functional profiling as well. Um, and some of the challenges here, not surprisingly, this is a high power technology. It often traditionally has higher costs and slower turnaround time. It also requires a lot of laboratory and informatics expertise to process the data. And it hasn't really been standardized yet for avian operations, although that's something that we're working on today. So, how should we think about integrating a proactive biosurveillance approach into our management system? In my title, I kind of answer the question. I don't think it replaces existing tools and techniques. We should continue with our observational health monitoring and our standard biosecurity practices. I think of proactive biosurveillance as really complementary to those existing tools. I would argue it's really kind of our first line of defense, right? What you're hoping to do with a proactive approach like this is really, getting an earlier detection of things that are in the barn already. The earlier you detect and diagnose, the earlier you can intervene, and hopefully you get better outcomes. Um, I think one of the kind of key areas for future development is this kind of proactive surveillance really sort of lives in a regulatory gray area for the most part now. There are some guidelines, for example, around salmonella testing in layers where we use boot socks and do this kind of regular testing. But there really aren't any comprehensive guidelines yet in the regulatory space. And so, one key thing you can do here is follow up any suspicious results that you get from a proactive biosurveillance system with confirmatory testing and standard diagnostics. And this sort of then funnels you directly into kind of, standard regulatory or reporting requirements, pipelines where there's real clear guidance on what to do if you get a positive hit with a traditional diagnostic. Um, I'm gonna well, I guess I can take a second here with this. This is just kind of wrapping up the section about kind of, what the future state looks like. And I kind of highlighted this at the top. I think by adding in this kind of proactive biosurveillance, you have a real opportunity to detect early, intervene earlier, and hopefully have better outcomes. So just kind of quickly, how does Barnwell do this today? What we're working with is, as I said, we use boot sock swabs. We put together kits that will ship out to a farm with everything you need to do a boot sock collection and all the details you need. So it's a relatively easy process. Anyone walking through the barn can do it. Those samples get sent to the lab and we apply metagenomic sequencing. So again, we get that full comprehensive picture of anything leaving DNA in the barn. We run our proprietary bioinformatics analysis and interpretation, and then we return those results to our customer dashboard so you can see as quickly as possible. And in our case, we have a 1 week turnaround time that we've worked really hard on. So within a week of shipping samples, you can start to see results of what we're seeing in the barn. I wanted to show a quick case study around this. So this is an example of working with a layer customer in the Northeast US, large operation with 2 million hens. We were doing continuous monitoring on 3 of their cage-free houses with about 25,000 birds per house for around 6 months. We were doing weekly sampling as we went along. Generally, kind of the take homes here, what happened? One of the houses had a spike in Candida, and this occurred shortly before they actually saw symptoms. of crop mycosis, and Candida is known to be one of the primary causes of crop mycosis. So key outcomes from this study, we were able to demonstrate that we could detect up to 3 weeks in advance and so give kind of an early warning for a risk of crop mycosis. Our system was actually comprehensive, so we were able to kind of detect and monitor everything, and I'll show that on the next couple slides. And this was able to inform the operations manager on kind of not only looking for disease, but looking for what their treatments were doing in the house, and maybe modify some of the treatment plans. So getting into this just a little bit more specifically, here what I'm showing is a graph of the relative abundance of Candida in their barn over weeks 14 to 37, so part of a layer house grow out. And so what you'll notice is around week 19, we saw a spike in the relative abundance of Candida. And this came about 3 weeks before they started observing symptoms of crop mycosis. As soon as symptoms were observed, they actually went ahead and treated with 1 pound per ton copper sulfate. And they ran that treatment for 3 weeks. And as you can see, interestingly, right after week 19, that spike, we already saw the relative abundance come down significantly, though not all the way. And then the treatment seemed to kind of knock it down the rest of the way, which was a good sign that the treatment was doing what it was supposed to. But interestingly, from the same sample, we're doing a complete microbiome monitoring, and we were able to see with the full microbiome that it shifts significantly as soon as the copper sulfate was being added in. And so what you'll notice here is there's kind of a large shift in the relative abundance of lots of different genera of bacteria. And you'll have to kind of take my word for it. But we did a deeper analysis on some of this data, and what we found is that a lot of bacterial genera that are associated with higher production outcomes that were present prior to copper sulfate began to go down in abundance. And simultaneously, we saw an increase in abundance of things that are sort of traditionally associated with lower production outcomes. And so, as I said at the top, for our operations manager, she found this to be really useful data because it sort of suggested that while the copper sulfate was doing what they hoped and treating crop mycosis and knocking down Candida, it might also be having some deleterious effects on the overall environmental microbiome health, and might impact production longer term. And so, I think what she was thinking in the sort of next round where she might have to do something like this, she may lower the dose of copper sulfate she was going to use, or possibly treat for a shorter amount of time to see if she could get the effect she wanted, but without these sort of secondary effects that might be negative to long-term production. One last thing to note here. So far, the Barnwell Bio platform is set up to monitor all DNA pathogens, but we've been working really hard to be able to monitor RNA viruses, which are also, there's a class of key pathogens, including high-path AI and avian metapneumovirus that are RNA-based viruses. And so we have been working really hard on figuring out the best way to stabilize samples that we collect in the barn, and then to optimize our laboratory processes so that we can start producing good, high quality RNA data and add that to our platform. So that's coming soon. Just want to finish up by thanking the entire Barnwell Bio team. And this image shows kind of most of the team here. I want to thank Dr. Stephen Rickey's lab at the University of Wisconsin and AgriGrow, who co-sponsored this symposium at PSA. And also thank the Foundation for Food and Agricultural Research, FAR, who sponsored some of our RNA work that we're doing. Thank you.